All right, let's dive deep inside our cells to follow a single, pretty remarkable molecule that you've probably heard a ton about recently. It's at the very heart of a medical revolution. We're going to track the ultimate messenger, RNA.
So our story really kicks off with a Nobel Prize that, and I'm not exaggerating, literally changed the world. Now, to understand how these game-changing vaccines work and why this award was so incredibly important, we first need to get to know the messenger molecule that makes it all possible.
So, here's the plan. We're going to follow this messenger's journey. We'll start with its Nobel-winning role, then see how it's actually made and edited inside our cells. We'll look at how it's all controlled, and then we'll zoom out to look at its incredible past and its even more exciting future. Okay, so what was the big hurdle here? For decades, scientists knew RNA had all this potential, but just injecting it into the body? That was a total non-starter. You see, not only is it incredibly fragile, but our immune system basically sees it as a foreign invader and launches this massive, really dangerous attack.
And this is where that Nobel-winning discovery was a complete game-changer. These researchers figured out that by making these tiny chemical tweaks to the letters of the mRNA molecule, they could basically create a stealth messenger. It could sneak right past the body's defenses, deliver its instructions, all without triggering that dangerous inflammation. It just blew the doors wide open for mRNA vaccines. So what exactly is this messenger, and what's its normal day job?
To really get what RNA does day to day, we have to start with the fundamental information flowchart of life itself. A process so central to biology, it has its own famous name. And that's the central dogma of molecular biology. Think of it like this. Your DNA is like a priceless collection of master recipe books. They're locked away for safekeeping in the library of the cell, the nucleus. You'd never ever take those precious master copies out into the chaos of the kitchen, right? No way! Instead, you'd make a temporary, working photocopy. Well, that disposable photocopy? That's messenger RNA.
But here's something that might surprise you. The messenger RNA, the one we hear about all the time, is actually the rarest type in the cell. It's only about 5% of the total RNA. The vast majority is ribosomal RNA, which actually builds the protein-making factories themselves, and transfer RNA, which is responsible for bringing the right ingredients to those factories.
So how do you make that RNA copy from the DNA blueprint? Well, that process is called transcription, and it basically happens in 3 main steps. You have this molecular machine, kind of like a biological scribe called RNA polymerase. First, it has to find the start of the right recipe in that huge DNA library. Then it scoots along the DNA, copying the code letter by letter by letter. And finally, once the recipe is complete, it hits a stop sign and releases the brand new copy.
But, and this is a huge but, for complex organisms like us, the story is far from over. That first copy that our little scribe makes, it's just a rough draft. It is not even close to being ready to send out of the nucleus. So is that first copy ready to go? Absolutely not. No way. In what we call eukaryotes, that's organisms with a cell nucleus, like you and me, that initial transcript is called pre-mRNA. And before it can be sent out to the protein factories, it has to go through this really rigorous multi-step editing process. Think of it like a post-production process. It involves 3 key steps. First, the mRNA gets a protective cap on the front, kind of like a little helmet. Then it gets a long, stabilizing tail on the back end. Both of those are there to protect the message from breaking down. But the most incredible step, honestly, is the one in the middle, splicing. You see, it turns out that initial RNA copy is full of, well, junk, non-coding gibberish. These sections are called introns, and they interrupt the actual instructions. So this massive molecular machine called the spliceosome comes in, and it acts like a master film editor. It finds the start and end of all these junk sequences, precisely snips them out, and then splices the important parts, the exons, back together to create the final coherent message. It's amazing.
Now here's where it gets absolutely incredible. The cell can splice that same rough draft in different ways. This is called alternative splicing, and it is a huge source of our biological complexity. By mixing and matching different exons, one single gene can create many different proteins. For instance, there's a gene that in the thyroid produces calcitonin to regulate calcium, but in your brain, that exact same gene is spliced differently to produce CGRP, a peptide that's involved in migraines. One gene, 2 totally different jobs in 2 different parts of the body.
Okay, so we know how the message is written, and we know how it's edited.
But wait a minute, how does the cell even decide when to make a copy in the first place? How does it turn genes on and off? Well, bacteria have these super elegant systems for doing this. They often group genes that work together into these functional little packages called operons. A classic example is the lac operon in E. coli bacteria, and it works just like a smart switch for digesting sugar.
The logic is brilliant and really simple. If the bacteria's favorite food, glucose, is around, the switch for digesting a different sugar, lactose, stays firmly in the off position. Why waste the energy?
So how does it stay off? Well, there's this thing called a repressor protein that literally binds to the DNA and acts as a physical roadblock. It just blocks the gene copying machinery from getting started.
But when lactose is present, the lactose molecule itself acts like a key. It binds to that repressor protein and pulls it right off the DNA. The roadblock is gone, the switch flips on, and the cell gets to work making the stuff it needs to digest that lactose. It is a beautifully simple, self-regulating system.
So, as we've seen, RNA is a master messenger. But honestly, that's just scratching the surface of what it can do. This is a multi-talented molecule that's right at the center of some of life's deepest, most fascinating mysteries.
For starters, get this. We used to think only proteins could act as enzymes, but we now know that some RNA molecules, called ribozymes, can actually catalyze chemical reactions all by themselves. Then there's the crucial enzyme telomerase. It protects the ends of our chromosomes and is linked to aging. Well, it uses an RNA template to do its job. And of course, whole families of viruses, from influenza to HIV, don't even have DNA. Their entire genetic code is written in RNA.
And all of this incredible versatility, the ability to both store the information like DNA and do the chemical work like a protein, has led to one of the most compelling ideas about the origin of life itself, the RNA world hypothesis. This theory suggests that way back, before the whole complex system of DNA and proteins ever evolved, life on Earth might have started with just RNA, a single molecule that could do it all, store the code, and build itself.
So you think about it. From its potential role as the actual spark of life to saving our lives in the modern world, the messenger's journey is just incredible. It started as a simple copy, and it's become a tool, a history book, and a key to our future. And that leaves us with the big question: what are we going to ask this incredible molecule to do for us next? Thanks for joining this explainer.